everybody, today we discuss the synthesis of cholesterol. Now cholesterol synthesis is a very long pathway, but we do it in a very easy way so that you can always remember. First we divide it into three parts. First is the formation of mevalonate. Second is the formation of isopentyl pyrophosphate, and third is the formation of cholesterol. So first is the formation of mevalonate, second is the formation of isopentyl pyrophosphate, and third is the formation of cholesterol. The site for cholesterol synthesis is in cytoplasm. So we see one by one. The most important step for forming cholesterol now will be the formation of mevalonate. So we see how mevalonate is formed. The initial synthesis of cholesterol, the first molecule that takes part in the formation of cholesterol synthesis is acetyl-CoA. But here we require 2 acetyl-CoA, which will form acetyl-CoA. So 2 acetyl-CoA molecule, when they combine, they form acetyl-CoA, and because there is only one CoA, so one CoASH is removed. The enzyme is thiolase. Then this acetyl-CoA combines with one more acetyl-CoA. Again a CoA molecule is removed to form beta methyl gluteryl CoA, or in short hydroxymethyl gluteryl CoA synthase. The enzyme is HMG-CoA synthase. So beta hydroxymethyl gluteryl coenzyme. Till now we have used only acetyl-CoA. Now again we wanted to form mevalonate. So the last step is formation of mevalonate. Now how is this mevalonate formed? This mevalonate is formed in the presence of HMG-CoA reductase. This formation of HMG-CoA reductase to form mevalonate uses energy. 2 NADPH plus H plus to form 2 NADP plus, and since the mevalonate now doesn't require any coenzyme A, so CoASH is also removed. So we wanted to form mevalonate first, and we have formed mevalonate through only acetyl coenzymes A. So it is very easy to remember. First 2 acetyl coenzyme A is used, then it forms acetyl CoA. Again one more acetyl coenzyme A is used. It forms beta hydroxymethyl gluteryl CoA. Finally mevalonate is formed. The enzyme which is most important in cholesterol synthesis is HMG coenzyme A reductase, and this reaction is the rate limiting reaction, or the rate limiting enzyme for this whole cholesterol synthesis is HMG coenzyme A reductase. So the regulation of this cycle is basically only on HMG coenzyme A reductase. So we have formed mevalonate. The first part of the cholesterol synthesis has been completed. 2 acetyl CoA forms acetyl CoA, which forms beta hydroxy beta methyl gluteryl CoA, and which forms a mevalonate. Now this beta hydroxy beta methyl gluteryl CoA, these reactions till the formation of beta HMG CoA is similar to the formation of ketone bodies, but the only difference is that this all takes place in cytoplasm. Okay. So this all synthesis is taking place in cytoplasm, and the rate limiting enzyme is HMG coenzyme A reductase. Now we have formed mevalonate. and from mevalonate we will form isopentyl pyrophosphate. Now when we formed mevalonate was a 6-carbon compound. Mevalonate is a 6-carbon compound. This mevalonate will combine to form, now we keep on adding phosphate group. So it will form 5-phospho mevalonate. This again we add one more phosphate molecule. It will form 5-pyrophospho mevalonate. Again we add one more phosphate group. So now it forms 3-phospho 5-pyrophospho mevalonate. Now finally there is a decarboxylation and it forms isopentyl pyrophosphate. Now why I didn't write the enzyme was because whenever the phosphate group is attached, the enzyme you should remember is a kinase. So this is a kinase, this is a kinase, this is a kinase, this is 6 carbon, this is again 6 carbon, this is again 6 carbon, this is again 6 carbon. The difference being that this is 5 carbon now because this enzyme is a decarboxylase, a carbon dioxide is removed. So the name of the kinase will be, this will be mevalonate kinase, this will be phosphomevalonate kinase, this will only be kinase or it will be 5-pyrophosphomevalonate kinase, and this would be phosphomevalonate decarboxylase. So now we have formed what we wanted to form was isopentyl pyrophosphate. Basically mevalonate was a 6-carbon compound. It had no phosphate group, but on action of 3 kinase 1, 2 and 3 kinase, it got 3 phosphate groups. One was 5-phosphate, then was 5-pyrophosphate, then was 3-phospho 5-pyrophosphate. So still it was 6 carbon, but carbon dioxide was removed in the presence of phosphomevalonate decarboxylase to form isopentyl pyrophosphate. So it is a 5-carbon compound. Now the important thing is, whenever you add phosphate you have a kinase, and whenever you have a kinase enzyme, ATP is utilized, and whenever ATP is utilized there is a mineral that is called as magnesium. So in every step you have to write ATP, ADP and magnesium. Kinase, the same reaction for hexokinase and glucokinase in glycolysis cycle. There also ATP was converted to ADP, and similarly magnesium was used up. So now we have done the second step for cholesterol formation, that is formation of isopentyl pyrophosphate. So first step was two acetyl coenzyme A molecules which were forming acetyl CoA, then HMG CoA, then mevalonate. This mevalonate was a 6-carbon compound. Now we wanted a 5-carbon compound so that we could form cholesterol easily. So it took this mevalonate underwent 3 kinase reactions where each phosphate group was added, and finally a decarboxylation reaction to remove a carbon dioxide and to form isopentyl pyrophosphate. So we have done the second part of cholesterol formation. Isopentyl pyrophosphate has been formed. Now coming to one mnemonic for the next part of the reaction. Now we have formed isopentyl pyrophosphate. So it will start with isopentyl pyrophosphate, which is 5-carbon compound, and as we know, cholesterol is the last step in this cycle that we are supposed to form and it is 27 carbon. So there are steps where cholesterol will be formed, that is 27 carbon molecule will be formed from isopentyl pyrophosphate, that is a 5 carbon molecule. Now the mnemonic, if we can remember after this, is D, the, GF. The is for D, GF, SLC. Okay, D, GF, SLC. The sequence is not supposed to be changed because we cannot take the risk of losing anything in this cholesterol metabolism. So D, GF, SLC, or the GF, so looking cute. You can come there, the GF, so looking cute. Okay, so isopentyl pyrophosphate. So D, from D we have dimethyl allyl pyrophosphate. Now this is a 10 carbon, this is not a 10 carbon, this is again a 5 carbon compound. This is formed from isopentyl through a isomerase. Because it is formed from isopentyl, so it is isopentyl pyrophosphate isomerase. Generally the name of the enzyme is either, the initial part is either from the reactant or the product. So this is isopentyl pyrophosphate isomerase and it forms dimethyl pyrophosphate. Now what will happen is dimethyl allyl pyrophosphate and isopentyl pyrophosphate will both combine. So 5 carbon plus 5 carbon will form 10 carbon compound, and now it is a turn of G, that is geranyl pyrophosphate. The enzyme is cis-prenyl transferase. Okay. Now we have formed 10 carbon, but we need to go to 27 carbon. So now is the turn of F. So geranyl pyrophosphate will again combine with isopentyl pyrophosphate. So 10 carbon over here, 5 carbon over here. So 15 carbon compound, that is farnesyl pyrophosphate. Again in the presence of cis-prenyl transferase. So now I have 15 carbon compound. This is one molecule. But now if I take 2 farnesyl pyrophosphate, means now I have taken 2 farnesyl pyrophosphate, 2. So 15 plus 15, I have a 30, I can make a molecule which is of 30 carbon compound. And that, what is that molecule? So the 30 carbon compound molecule is squalene. Okay. So 30 carbon compound molecule is squalene, and then is the time of L. So L is lanosterol. Lanosterol is again a 30 carbon compound. So 2 farnesyl phosphate pyrophosphate will form squalene. Squalene will form lanosterol, and lanosterol will form cholesterol. That is 27 carbon compound. First we'll see this much part. Isopentyl pyrophosphate formed dimethyl allyl pyrophosphate. It was a 5 carbon compound through isomerase enzyme. This 5 carbon dimethyl allyl pyrophosphate added upon with isopentyl pyrophosphate to form a 10 carbon compound, that was geranyl pyrophosphate. This geranyl pyrophosphate again added with isopentyl pyrophosphate to form farnesyl pyrophosphate. Now this was a 15 carbon compound. This was 15 carbon. So 2 farnesyl pyrophosphate combined together to form squalene. Squalene then formed lanosterol. So these both are 30 carbon compounds, but cholesterol was 27 carbon compound molecule. It was formed. Now we'll see from farnesyl pyrophosphate again how the cholesterol is formed. It is a lengthy cycle, but once if we remember forever. Now coming to farnesyl pyrophosphate. farnesyl pyrophosphate, that is 15 carbon compound. So 2 molecules of farnesyl pyrophosphate would form squalene, and again it is an energy consuming process. So NADPH plus H plus consumed, NADP plus released. Now this requires Mg plus 2, Mn plus 2. Now this squalene will form lanosterol, and again this will require as energy consuming process NADPH plus H plus, NADP plus, and one oxygen is also required in the process, and lanosterol is formed. But still it is 30 carbon, and still it is 30 carbon because carbon is not being removed. Now through series of reactions around 19 cholesterol is formed. That is 27 carbon compound, where 1 carbon is removed in the form of formic acid HCOH, 2 carbons are removed in the form of 2 carbon dioxide. Again energy consuming process, NADPH plus H plus is required, and the enzyme and this is about series of 19 reactions from lanosterol to cholesterol. Okay. Finally, with the removal of 3 carbon compounds, 27 carbon compound cholesterol has been formed. Squalene to lanosterol, the enzymes are epoxidase, hydroxylase, and cyclase. So 2-farnesyl pyrophosphate forming squalene, forming lanosterol, and forming finally the cholesterol. Now if I say the last 3 series of our reactions are the ones which use energy in cholesterol formation, that is from farnesyl to squalene, squalene to lanosterol, and lanosterol to cholesterol. So these 3 are using NADPH plus H plus, and the one initially which was the rate limiting enzyme was using NADPH plus H plus, and all the kinase were using ATP. So the energetics is not that difficult if we remember that from farnesyl all the reactions require NADPH plus H plus. The HMG coenzyme A reductase reaction requires NADPH plus H plus, and all the kinase reactions require ATP. So there are 3 kinase reactions that are requiring ATP. Okay, so this is the formation of cholesterol, and as we have told that HMG coenzyme A reductase is the rate limiting enzyme or the main enzyme through which the cholesterol cycle is regulated. So HMG coenzyme A reductase is the enzyme. HMG coenzyme A reductase is the enzyme which is inhibited by the drugs, lipid lowering drugs, that is statins, to decrease the formation of cholesterol. HMG coenzyme A reductase was the enzyme which formed from HMG CoA, it formed cholesterol. Cholesterol itself reduces the synthesis of HMG coenzyme A by hampering the transcription of this enzyme HMG coenzyme A. Now this enzyme HMG coenzyme A is stimulated by insulin and thyroxine. Insulin and thyroxine, and the other hormones which are antagonists to thyroid and antagonists to insulin will inhibit HMG coenzyme A reductase. There is one simple thing that we can remember: in fasting state all the enzymes phosphorylate, but here we do not want the active form of HMG coenzyme A reductase is dephosphorylation. So insulin and thyroxine will dephosphorylate HMG coenzyme A reductase. Thank you.